Hi everyone. So let's look at the molecular biology behind this experiment now. So at this stage, right here, when the hormone was being added to the particulate fraction, now remember that the particulate fraction is a fraction that has the membranes in it. And membranes have what? They have receptors. Right here, I have that mentioned here. So membranes have receptors, and receptors are the key players in this experiment, right? So in the fraction that contained the membranes, the particulate fraction, when the hormone was added, this hormone basically acted as a ligand. When this acted as a ligand, it bound itself to the receptor and it did the whole thing that we went over a few slides ago. So it activated the G protein, G alpha subunit got activated, transferred its converted GDP to GTP, and then went about activating the effector, which because we know it, which in this case is adenylyl cyclase, and then when adenylyl cyclase is activated, it produces cyclic AMP. So basically, you have started the production of cyclic AMP because of that hormone acting as a signal, okay, as an extracellular signal. So when they mixed it really well, you have a lot of cyclic AMP at this point. And when you add to this solution that had the cyclic AMP molecules, when you add the supernatant to it, you see reaction, because cyclic AMP is the enzyme that is responsible for activating phosphorylase. Okay, so let me just mention that on the next slide, so it's clear. Is there stuffed in space there? Oh yeah, there's space here. We can just write it here. Okay, so cyclic AMP activates phosphorylase, and then phosphorylase is now active and can go about doing whatever it does. So we see activation. So we did not see activation of phosphorylase when we just added the hormone to the soluble fraction, although phosphorylase was present in here, right? So if the phosphorylase was present in here and you add the hormone, it didn't really do anything, because you need that signal to be transduced first, and you need to produce cyclic AMP before you can go about activating your phosphorylase. Okay, hopefully this has made sense, because this is sort of it's a little, it took me a lot of time to understand this experiment, so I wouldn't blame it. So don't get too worried if you don't get it in the first go. It just takes a little bit of time to understand this. Okay, and I'll just, since we have 2 minutes left, I'm just going to read out this note from my notes. So Sutherland identified the substance released by the particulate fraction membranes as cyclic adenosine monophosphate, or cAMP. This substance was released by particulate fraction into wash and activated phosphorylase. Cyclic AMP activates glucose, well, cyclic AMP then goes about stimulating glucose mobilization, which further activates a protein kinase that adds a phosphate group to a specific serine residue of the phosphorylase polypeptide, and then that activates your phosphorylase. Okay, cAMP is the second messenger. It is released into the cytoplasm due to first messenger, could be hormone or any other ligand, binding at cell outer surface, and diffuses to other sites in the cell. Okay, what I have next is again a picture from the CARB textbook. So I'll just quickly mention that. CARB, 5th edition. Okay. So what you see in this picture, and let me see if I can, 1 second, increase it a little bit. Here, there we go. That's better. So what you see in the picture, what it says here is localization of cyclic AMP in a live cell in response to an extracellular messenger, the neurotransmitter 5-hydroxytryptamine. So that is another ligand that it can respond to. So before HT was added, this is what it looks like, but as soon as it was added, so this yellow stuff here is all cyclic AMP. So see how it is, produced as a result of the neurotransmitter binding, and it is found at very, very specific locations. It's not everywhere, at very specific locations. Okay, so that's the whole point of this picture, and in the next video we'll continue on with something else, because this video is ending. See you guys in the next video.